And maybe kind of we will ask one or two of the questions that were directed more to Adam himself, and then we will just continue for the overall discussion. So I will pick up one question that is there, and it's really awesome work, and I join this time. And I assume that dopamine is coming from the VTA. Do VTA dopamine dopamine neurons express the IL-6 receptor? And do you think it's actually IL-6 is driving that? And you kind of referred to a code, so maybe you can kind of expand on this whole idea.
Yeah, I mean, the code I meant for more the acute things when there's sort of an injury and there's something to be signaled. I think in these chronic inflammatory states, it's a little different. There's sort of convergence of factors. Now, the running idea in this field is that a lot of things happen in the striatum itself, that it's a local process there where dopamine is modulated. So we don't think so, because we also measure dopamine neuron activity. So the dopamine neuron activity is itself impacted. We don't know if it's direct or indirect, but it's certainly pointing us to a neural circuit explanation, as opposed to sort of a distributed explanation with sort of local astrocytic processes changing release and so on.
So I guess it also answers a little bit the question of whether the pro-inflammatory cytokines, where are they produced? Are they produced in the brain as well?
We don't know. We don't know. I mean, it's actually very interesting. So IL-6 goes up in the periphery early and really only gets high after the onset of cachexia. And there's also blood-brain barrier disruptions then. So we don't know if the blood-brain barrier disruptions are probably indirectly, at least, if not directly, driven by IL-6, and that's how IL-6 gets in, or that's also sort of an amplification process. But we can block this with the IL-6 antibody peripherally.
And then, well, I guess it's really eventually, Michal is really missing here to put the connection there of how the peripheral IL-6 can also change the overall environment. So then maybe let's start the overall discussion. And I think what came up from both of your talks, and I would say also from Michal's talk, is this idea that we keep using this mapping, right? We keep using this mapping as a way of, you use this code, cytokine code. Carlos is using this idea of kind of an atlas representing activity. And it kind of begs this homunculus idea that there are many different homunculi, right, that represent that. So maybe, what do you think we're going to find there? Like, what are we looking for, how they interact, and what do you think we're going to find there? Maybe let's take it to Carlos and then back to Adam.
Yeah, the way I think about it is that that's how science often starts, right? I mean, you first start by mapping out things and transforming one thing from one space into the other, right? From one language into the other. But it's true that often this kind of still leaves, I mean, I have this slide I use for teaching, right? From Men in Black, for the ones who are old enough to remember that, right? Where the skull open and there's an alien in there, right? And I always say, well, how many times do we need to open that? I think that we are now entering exciting, or we are already in the middle of exciting times, because we're going now beyond this homunculus idea, right? Really thinking about how is information now being used to then really activate different motor output patterns, right? Or how can we really find and explain ways using theory. That's another way people are approaching this, right? To explain maybe some computation, some higher order computation of how this is working. Now, obviously, we don't know how to do it, because if we would know how to do it, we would know how to solve the brain, right? But I think that there are these steps, right? There's the stamp collection stage, there is the atlas building stage, and then there is the dynamic stage, where you start looking at how these things change around, and then you go in and try to see if you understood the language, right? And so in my view, this is how things progress. And we are definitely now in the stage where we're going beyond the homunculus, and I think that's exciting. But you cannot get there without making the stamp collection and the atlas part. I don't know how Adam thinks about that.
Yeah, I don't think I would call it a homunculus. I think we're just searching for the regions that we're not sort of aware of at this resolution. And I think that's just a mandatory first step before we can really understand the computations and what's happening. I also think it's just very exciting to sort of see different fields come at the same problem from very different perspectives. So when I started getting into this and trying to read about cytokines, it's just so interesting how neuroscientists would approach this versus an immunologist. And I think it's just going to be very exciting. And I wonder if immunologists might think the same thing about what we do in the brain as the very first, strange first thing to do compared to what they would have done. But I wouldn't downgrade sort of these mapping results as homunculus. It's just really the first steps.
And then it seems that motivation, it also comes up in the question, right? Motivation appears in everything there, in your in every possible work. And then I guess the question there is what's going, like, why motivation changes when you show in sickness behavior, you show the motivation changes. So in one case you can see that they are more motivated to feeding or heat or something else. So the motivations are very different, and I think it also speaks to the whole idea that if there is a code that controls motivation, what is the structure, or what should it integrate in this sense?
So Jeremy Borninger is also asking a similar question about the energy constraints of mounting an immune response. And I think it's a very related question to what you're saying, Asya. I think that's what we have a brain for, is to figure out the cost benefit kind of scenario. I don't think that's in the code. I think your sense, sort of the brain senses the periphery and the pathogens and the organs through these mechanisms. But the brain has to decide whether the sort of the cost of mounting an expensive operation outweighs the benefits. And there's not an easy answer, and I think that's why we have a brain. So I do think once we get to those kind of interesting questions, we are sort of back into systems neuroscience where I feel really comfortable.
And maybe I think there's an additional way of thinking about it, right? Like, in case the body does its stuff because it really needs to do it, the brain is left having to deal with the consequence and find a solution of how to deal with that, right? So it's kind of the, right? Because I think, I mean, evolutionary speaking, for me, it's fascinating. I like to really read a lot of microbial literature, right? Like, how do microbes predict the future, right? So because they can do that, right? So there's this fascinating study where gut microbes, when they get into, so what that happens, if you increase the temperature a little bit, they will actually start expressing pH pump, which make them acid resistant, right? And so the idea why that happens, because when they get into your mouth, they have evolved to know that in a few minutes they're going to be in a very acidic environment. So they better start actually already now expressing these predictive things, right? And a bit that's how the brain does it, right? And sometimes there's no choice, right? You need to act, and you need to, the organs need to do stuff. And that's an internal thing. Now, what the brain can do is it can change how the whole organism interacts with its environment, which obviously organs cannot do that. So it has to take this big picture decisions of how to navigate the world according to what the internal world is. So kind of the way I think about the nervous system is that it evolved to deal with a problem that individual cells within an animal are not independent enough, right? Are not independent anymore. And so now you need someone else to take these big picture decisions.
So I think it's maybe, Carlos, it will take us to this the question that keeps coming back. If you can explain protocognition, and I think from there you can also answer the question that many people ask: flies?
Yeah, well, so there's two ways of thinking about why I like to use the word protocognition, because again, I think that cognition is very often associated with really higher order cortical type of computations, right? Creativity, things like that, which obviously it is associated with, right? But or neuroeconomic decisions, right? Should you buy the car today or should you get a loan or whatever, right? Now, we haven't evolved to deal with money, right? We haven't evolved to make decisions about mortgages. So where does this come from, right? So what's the what's the original building blocks to think about these computations? And as Adam was saying, it's about dealing with limited resources, right? It's about making these trade-off decisions. And honestly, again, unicellular organisms had to make these decisions, right? Do I invest into expressing transporters to get more nutrients in, or do I invest into making a new cell, right? And so somehow I feel like many of these, and if you scale this up, obviously now into really making predictions over larger time scales, or using information which evolutionary hasn't existed, but only exists within the time life of that organism, I think then you start getting into this more brain cortical type of cognitions. But the origin of that is to deal with two resources, food and mates, right? And they're not easy, as Adam was saying, they're not easy decisions, right? So how to really deal with all these complex loops. And so I don't think you need to go very far into human cognition to start understanding the building blocks for these computations. They must exist already very early on in life. And obviously invertebrates, flies, C. elegans, I mean, bees are amazing, right? I mean, what some insects can do, or ants, is amazing. So they must have evolved early on. If not, they wouldn't be around. And so we can get insights into these things by studying these numerically simpler brains, and then get the real mechanistic insights to then look into that in humans, for example. So that's the other way of thinking about it, is that if I write cognition and flies in a grant, it won't fly. So I use protocognition to not piss off anybody. That's another way of answering that question.
That's a good one. So actually it brings us to, I think, to another level, and it's relevant very much to what Adam was talking about and what you're talking about. But many of the decisions eventually that we do, including sickness behavior, including nutrition, are completely cognitive, and they are affected by states of mind, right? Or the way you perceive cognitively a certain state, you can almost switch the system. Or to how do you think that is happening, or to what extent it's really happening? Maybe Adam?
I mean, we don't know, of course, but I think it's almost sort of a question of perspectives. I think if you're thinking about it as sort of what's in neuroscience called a labeled line code, where you have sort of one cue wired to one response, that sort of might not be the way to think about it. Or if you think about sickness behavior as just one entity, that may be not the way to think about it. But if you think about the process more as a new sensory system that's kind of been unexplored, that provides a rich set of information, then now you can start to think about this issue the way a lot of other sensory systems are thought about, which is, yes, there's standard transformations that happen, there's typical patterns that they can elicit, but it's context dependent, and we can kind of start to probe the conditions when sort of different ones get integrated, where there's cost-benefit analysis. To me, it's really kind of a perspective, but that, again, comes definitely from my systems neuroscience background.
Carlos?
Yeah, I totally agree with Adam. I think that one can start thinking about interoception as just another way of doing sensory information. And then you can start thinking about mapping these like in sensory space, right? Doing psychometric curves, right? I mean, when we play around with the different nutrients in the diet of the fly, you can think about it as a psychometric curve, to think about it again as a system neuroscientist, right? I think to the question you were also asking about these sudden switches in perception and like mood and so on, I think that's a fascinating question about what are internal state, something which more and more people now use. It's also system neuroscience, right? But we know that if you look at brain activity, there are these switches which happen, right, with pupil dilation and so on, with being also part of that. And so to which extent these circuit dynamics and changes are these, how they relate to internal states or not internal states, or do internal state, like, sorry, physiological states, tap into these dynamics and so on. I think that's a fascinating current discussion within systems neuroscience, which we do not understand. And I think the dialogue there for me is really fascinating, right? Like an organ state, a transcriptional state, what's the real difference to a brain state, right? So I'm a trained molecular cell biologist, right? So I like to think a bit about fundamental principles controlling these things. Is there something fundamental different about how the brain does it, or like what would be like a liquid state machine at the level of the liver, for example, right? So I think there's a lot to be discussed and discovered and identified by these discussions across different fields.
Do you think we are ready to answer those questions? You think?
I think we're ready, because the molecular tools, cellular tools we have been using for a long time now can be actually implemented and dynamically controlled with precision within organisms. It's a high dimensional space. So what to really manipulate is the question. As Adam was saying, cataloging is really important there, right? But I think that we are ready for it. And again, if you think about it, fields in biology have always been doing that. Developmental biology, right, is a systems question, right? You cannot look at the development of a single cell in a certain state, right? Immunology at the whole organism level has always been asking this question. Physiology, you can argue that what we're discussing is just the 20th reincarnation of physiology as a science, right? Like 10 years ago, it was called systems biology, right? Now it's brain. It's the question of asking regulatory principles of living systems at the whole organism level, right? I think we are ready, and the progress of the field is showing that, and it's exciting moments also because of that at the moment. But it's challenging, right? And we often don't know how to do it. But that's why we need talented people working. Right? And the young people nowadays are amazing.
Yeah, absolutely. And Adam is kind of took us to this, I think one of the most challenging frontiers is psychiatry, right? And Carlos also mentioned that sensory systems might be altered in psychiatric conditions. Adam, how do you think we can approach this psychiatry in a more systemic level? Do you think it's basically, maybe it's the only way to approach psychiatry on the one hand, but on the other hand, what do you think kind of the gap that we have there in order to be able to answer that, to kind of start touching these questions?
That's a great question. But before, sort of one of the answers is that we now have the tools, which is also an answer to what the previous discussion you were just having, that yes, it might be a rehashing of what used to be called physiology, but it's not without merit, because now we can actually address things that we just couldn't before. And that's how I feel about this situation, is that for the first level, I feel like it's really immunology and other fields of biology that are providing us with models that have a psychiatric phenotype. So in that sense, it's looking in the brain of diseases where people just haven't looked in the brain yet. And again, cachexias, that's not only for cancer, right? There's a lot of other cachexias. And then we can start looking at convergences, right? So we know that chronic inflammation, even if it occurs in the context of, say, rheumatic arthritis or cancer cachexia, is very different at the bodily level. And yet they have shared behavioral phenotype. So that's a very exciting question, because it suggests a certain type of convergence. So I think that would be the sort of next step to think about this problem. And I think slowly I would hope that that takes us to to clinical depression, which, to be clear, I don't think it's the same thing, but we might get a handle. And in particular, right, in clinical depression, sort of probably the brain to body arc of this many loops might be more important. But I feel like until now it's been very squishy, at least for us trying to do psychiatry. And I think this really, really strong immune component, I think, really puts us in in front of incredible opportunities.
And Carlos, how do you think nutrition, like you mentioned that before in the context, is there evidence that kind of establishes nutrition as a strong factor in this disease?
Well, I always feel uneasy to talk about that given my training and my background, but definitely, I mean, there is a strong movement in so-called nutritional psychiatry. There is obviously a lot of interest in the association between microbiome, brain, and psychiatric diseases, thinking about the microbiome as a nutrient producing and modifying entity, right? Puts it maybe in that direction. Now, if you talk to like practicing clinicians, right, many of them are doubtful to which extent these things are really reproducible and impactful. They're worried also that, that this opens the Pandora's box to charlatans and also to people avoiding efficient treatment. So I think there's a promise, there is likely to be a biological background to it, but how to really implement that in clinical practice, how to do that in a very targeted way, because it's clear that it has to be personalized also, right? And to which extent it is treating epiphenomena, right? So, for example, most children with autism have digestive nutritional challenges. And so diet interventions are strongly associated with an improvement in well-being and in cognitive performance of these children. Now, are you just making them not feel sick and that actually helps then the brain? Or are you directly intervening at the brain level? We just don't know, right? And ultimately, right, again, think about brain-body interactions as a whole system. Does this really matter? Right? But it is a promising, complex question. And my personal opinion to this is the way forward is mechanisms, understanding how it really works, so that you have then a more objective molecular platform to then evaluate some of these things beyond the clinical epidemiological approach, which is obviously very important too. But doing prevention is even more complicated, right? Because in nutritional interventions over 10 years, that's hugely challenging.
So actually, in a way, maybe it all brings us to a question that I think is science will always look for causality. Maybe it's a way our enemy in this case in brain-body interactions.
I'm not sure I understand your question, Asya.
The way we usually look for causality, that something, this is the initiator and this is the outcome. But when we are talking about these systemic systems, maybe the fact that we are looking for causality, we're actually shooting our own leg. Maybe we're doing the wrong thing here. Maybe we need a different paradigm to study brain-body interactions, because causality is not unidirectional anymore.
Yeah, I think another way to ask that maybe is what's the limit between reductionism and systemic thinking about that, right? And I think that's also where I feel I've learned a lot from thinking about from system neuroscientists, right? Where at one stage you have to go okay, let's try to model that to start seeing things which maybe you cannot put so easy into a narrative perspective, right? And start using more modeling approaches to start understanding that. The question is obviously, if the model is correct, but you cannot make a narrative out of it, is this really understanding? But then we are really going into philosophy of science type of discussion, right? But I think I think there is value of using computational models when thinking about multi-dimensional, multi-feedback loop type of systems, which I think we can learn a lot from. I personally learned a lot from system neuroscientists thinking about that, from my colleagues here at Champalimaud, for example, who do that. Yeah.
So then maybe in the last 30 seconds that we have, Adam, do you think, like in the concept of neuroimmunology, where there was the idea that immune cells don't enter the brain, is there something like that that holds back the whole field for you?
I don't know if it holds it back, but I think when I was looking at the literature of this sickness behavior type, I think there's sort of no circuits in it. It's all cells across the body and the brain, but no circuits. So I think the new insight, and that's not new to a neuroscientist, is that the answer may be in sort of cell types connected to other cell types that we call circuits.
So then I think we will wrap it up, and maybe we will wrap it up. But by the way, we started the whole talk together with Limor in the beginning, when we were talking about between ourselves, who is in the brain-body field. And I think it's really everyone. And I think this is the most exciting part, that everyone are part of this field that seems that basically needs all this perspective to be formulated. So with that, I would like to thank you very much for your participation. There are several questions that remain, and Carlos, your really, it seems to be a very interesting direction for everyone. So there are some questions, and I will move then to Sarada, and thank you very much for joining us, and please stay tuned.
Thank you, Asya, and thanks to all our panelists today, Carlos, Michal, Adam, and of course, Asya yourself. Great talks, a very engaging discussion. A big thanks to our audience for joining us today and also participating with some very interesting questions. As a reminder, the recording of today's events will be available on our website and also on Janelia's YouTube channel. Like, give us a couple of days and then the recording should be up. We'd love to hear back from you as to how you're doing and to hear any suggestions that you might have for us. So please take a moment to complete the survey. Do join us next month on November 2nd for the next event. The topic is RNA therapeutics and is hosted by John Curtin School of Medical Research at the Australian National University.